Hello students, today I'll explain about the active immunity as well as passive immunity. On what basis? That means you can tell that on basis whether the antibodies are produced by our own body or it is externally antibodies are injected in our body. Based on that, there are two types of immunity, the active and passive immunity. So I shall explain this directly with the difference between active and passive immunity. Starting with difference of definition. So active immunity, difference with that of passive immunity. Difference with that of passive immunity. What is the difference between them? If you know the difference between them, you can also understand the two concepts clearly. And let me tell you, it is one of the important topic both in terms of NEET as well as theory. So what is active immunity? Active immunity here, again starting with a person is there and naturally a pathogen has entered. So pathogen has entered inside. So a pathogen is there along with its respective antigen will enter inside the body of an individual and encounters the immune system. So once it encounters, here the question is not whether it is entering first time or whether it is entering second time. That is not the question here. So what is the question is, when pathogen enters, who will produce antibodies? Whether the person's own antibodies will be there or ready-made antibodies will be there. So here, when a pathogen enters the human body, the human body will start producing Y-shaped molecules. These Y-shaped molecules are called as antibodies. Okay. Now we are not considering whether it is first time entry, second time entry, or subsequent entry. You are only seeing where the antibodies are produced. As you can see, the pathogen has entered this person. Okay. And it is this person who has produced antibody. That means it is his own antibodies. It means that suppose pathogen enters your body also, and if your body produces antibody, it will be your own antibody. In that case, it will be active immunity. This itself is the first difference. In active immunity, own antibodies are produced. Then, students, however, in the next after this, however, I'm going to cover up the remaining part of what I have explained after the structure of antibody. Since this is important, this topic has to be stressed upon. So, own antibodies are produced. Person's own antibody, his own B cells will differentiate into his own plasma cells and will produce his own antibodies. So, own antibodies will tell that the immunity type is what? Immunity is due to active immunity. Active immunity is called, not due to active. It's called as active immunity. See, on the other hand, passive immunity. For that, suppose a person, pathogen has gone inside again. Now, enough time is not there for production of antibodies. You know that in primary antibodies, 7 days it takes, and in secondary, in primary immune response, it takes 7 days, and in secondary immune response, it will take at least 3 days. Okay. Now, if the pathogen can cause the symptom, is very dangerous and causes the symptom very quickly, within the 3 days itself, then body is unable to produce its own antibody. So here, own antibodies won't be there, especially for highly pathogenic organisms. It can be rabies, it can be tetanus, or it can be snake poison. Okay. Suppose a person is bit by the snake, then the proteins present in the venom will start acting as foreign and triggers the various effects. So how to control? Those proteins of the snake venom has to be bound by antibodies so that those proteins are neutralized. But does a person have enough time after the snake bite? No, he cannot wait for hours also. Okay, but for our system to produce antibodies to secondary response itself, it will take at least 3 days. Now, in this case, like in emergency case, you can tell. In these special cases, as I already mentioned, tetanus, rabies, or snake bite, injection has to be given. So now enough time is not there here for production of antibody. So a person should be injected with ready-made antibodies. Person should be injected with ready-made antibodies. So now, when the injection is given, then these antibodies, usually intravenous, for example, within the venous, within the vein, the injection is given. These antibodies will be released within the circulatory system of the human. Okay. Now, it is not owned. It is ready-made antibodies which are present in the injections, which are produced in various pharmaceutical industries. Okay. So therefore, that is called passive immunity. It is not due to use own antibody, but it is due to ready-made antibodies, which are present in form of injections. So here it is ready-made antibodies. So this was one of the difference between the active immunity as well as passive immunity. The next difference is it will take some time. Okay, it takes longer time to act. Act means what? See here, for this pathogen is not so dangerous. So enough time is there, 3 days or more, enough time is there. So immune system will produce its own antibodies, and those antibodies will finally prevent this pathogen from causing a disease. Okay. But for this process to occur, definitely it will take minimum 3 days or more than 3 days. So it is slow to act on the pathogen. Okay. So it is slow to bring about immunity, or slow to act upon the pathogen and finally bring about immunity. Okay. So this one you see, passive immunity is not that. It will instantly, already ready-made antibodies are injected into the blood circulatory system. So instantly it will start acting. Okay. Starts acting, fast acting it is. Instantaneously. It will start acting immediately or instantaneously, or it is very much fast acting. So this is the second difference between an active immunity and passive immunity. The next is so the third difference, it is long lasting. Definitely our body produces antibody, it will last for longer time. So immunity lasts for longer time. It means the antibody can be continuously produced. It means the plasma cells which are formed from these cells, once they produce antibodies, they continue to release till the pathogen is neutralized. So they are long lasting. Okay. This type of immunity is long lasting. Okay, this type of immunity is long lasting. It means that that antibody will remain here. What is that first? The antibody will be continuously synthesized. This is one point. And these antibodies, since it is continuously synthesized, the antibodies against this pathogen continue to be remaining in this body for a long time. So it is long lasting. Whereas it's not long lasting. This is not long lasting. Why? The amount of antibody within this injection, ready-made injection given is fixed. Okay. So once those antibodies will do their work, they are going to be cleared from the system, clearance. And still if pathogens are there, then one more injection has to be given. That means a single dose may not be sufficient. May last for a very short time. Okay. So therefore, there might be a requirement that the second injections also can be given. Okay. So not long lasting. These antibodies which are there will be cleared. Okay. So as a result, we see that this immunity is not long lasting. So here, active immunity, last one, it is harmless. It does not cause any allergy because our own antibodies are there. So a person cannot be allergic for these antibodies. But remember, these antibodies present in an injection will be raised in animals. Okay, that means these are animal antibodies. Okay. So what do? They inject the animals with antigen, raise the titer of antibody in its, then isolate its serum and finally obtain the what? The antibody. When these antibodies are produced in animals, animal components are foreign to us because we are human beings. Animals belong to a different species. Then what happens? It becomes a foreign. In fact, many tissues from one individual to another individual cannot be accepted. Okay. So also here, if it is different animal only, definitely these antibodies will be considered as foreign. And another is, how is the preparation of it? After the injection is prepared, certain components of those substances present in the serum of those animals can be present here, and those substances can cause allergy. So it is harmful. So it might cause allergy for substances. For what substances? For the substances that are present in that animal in which the antibody has been raised or antibody has been produced. What? So this is the fourth difference between the active immunity and passive immunity. So active immunity further can be divided into two types, and passive immunity also can be divided into two types. So this was the difference, broad difference between an active immunity and passive immunity. Here, active immunity can be further divided into two types, whether the immunity is natural or whether the immunity is artificial. Based on that, again, active immunity can be divided into two types. It is natural active immunity. Natural active immunity. Another is artificial active immunity. So what is natural is pathogen will naturally enter. Suppose a person stays in environment and somehow pathogen enters and he will get the disease and he will recover from the disease. It means that a person can recover from the disease because he has produced antibodies against those pathogens. So that is the best example that can be given for natural active immunity. So this is natural active immunity. So what is natural active immunity? Recovery from a disease. If a person recovers from disease naturally. For example, there is one pathogen entered inside the body of an individual. He will get the disease, but what happens? His body also produces the antibodies. Those antibodies will neutralize the pathogen and he will slowly recover with the disease. So now you tell me whether the pathogen are natural or artificial. It is natural. So whether the antigens present on the pathogen are natural or artificial, based on this it is natural active immunity and artificial active immunity. Right? So that is the difference between. Okay, so first what was the division? Whether our antibodies are own or ready-made antibodies. Based on that it was active immunity and passive immunity. Now whether the pathogen is natural or artificial, based on that it will be active immunity natural and active immunity artificial. So artificial active immunity. Got? So here example is person recovers. Here what happens? His own antibodies are there, that's the active immunity. But pathogen is natural ones. Naturally he has encountered. He did not purposely inject any pathogen or something, intentionally injected or something. No. So it is a natural pathogen entry. So his own antibodies. So it is natural active immunity. So next one, artificial active immunity. Artificial active immunity. Now what is artificial active immunity is pathogen is injected purposely. Where it is injected? In case of vaccines. Vaccines are different, antibodies are different. Now what is vaccine? Here the injection has dead pathogen. Injection has dead pathogen, not antibody. The injection don't have an antibody, but it has a dead pathogen. Now that dead pathogen, when it goes inside, also immune system will consider it as foreign and start producing antibody. That's how our immune system functions. So even the dead pathogen, dead killed or attenuated forms, means dead forms, or which are not causing disease, means attenuation, means causing disease, but they have made it inactivate, virulent. Now these pathogens, when they are injected intentionally through vaccine, if it is injected, then these dead pathogens will go inside the body, and again immune system will start producing antibodies even against the dead pathogens. Okay, whose structure has been maintained, whose structural integrity has been maintained. Understand? So those dead pathogens will stimulate our immune system to produce antibodies. Again, here primarily all antibodies are there, that's why it is active. But the entry of pathogen is artificial, so it is artificial active immunity. Now, best example of artificial active immunity is vaccination. So several vaccines are given. What is vaccines? It is an injection. Okay. Vaccine is an injection given. Why it is given? so that person in future will not get the disease. So polio vaccine cannot be given to a person who has already a polio, but it is given in advance so that in future he will not get the disease. So the injection given in advance to prevent the future occurrence of disease is called as vaccine. It means that in advance an injection is given so that the person in future will not get the disease. He does not have a disease, but so usually vaccination is given in children as immune system is more active, and most of the T cells and B cells mature during development mature during the childhood. So most of the T cells develop and mature during the childhood. So this is called as vaccines. So vaccines is an example of artificial. Now here, even in passive, there are two types. Whether the ready-made antibodies are natural or artificial. Here, whether the ready-made antibodies are natural or whether the ready-made antibodies are artificial. So passive immunity, if the antibodies are natural antibodies, it is called as natural passive immunity. If the antibodies are artificial, then it is called as artificial passive immunity. So this is how what functions in the passive immunity functions. Okay. So examples. Once you know the examples, you will come to know. First of all, passive means ready-made antibodies will be there. Means not owned. Ready-made here means anything that is not owned in a common way to understand. Okay. So I shall give an example of natural passive immunity here. Natural passive immunity. In the previous class, I was explaining about the antibody that passes the placenta, and that is IgG. IgG is an antibody that will pass from maternal circulation, passes the placenta, enters into the fetus, and then protects the fetus. Okay, that is how. That means the fetus is getting the ready-made antibodies from mother. It is not producing its own antibodies. So therefore, it is a passive immunity. So here, example will be antibodies from mother to fetus. From mother to fetus. Antibodies from mother to fetus. This antibody can be called as what? Is it the own antibody? Fetus is own antibody or taking from mother? It is taking the ready-made antibodies from mother, which is a naturally, it is not intentionally injected or something. So it is a natural process. So it is example of natural passive immunity. Okay. So naturally the antibodies are passing from mother to fetus. The fetus is getting ready-made antibodies. Now second example also, which I discussed in the previous class while I was explaining about types of immunoglobulin. So one of the immunoglobulin that is present in colostrum will be called IgA. I told you even after the birth, the infant still requires immunity support from the mother, even after the birth. So because still immune system will be developing. So the colostrum has antibodies called antibody A that will provide immunity to the newborn, the neonate. Okay. So that means that is also an example of the newborn will get the neonate is going to get the antibodies through colostrum, ready-made antibodies, because those are the antibodies produced in the mother's body. So it gets. So here it is antibodies present in colostrum. Present in colostrum. Okay, so this is the examples of natural passive immunity. Next one is artificial passive immunity. So artificial passive immunity, giving example of it will be injections. I already gave an example, ready-made injections. That means this is an artificial, it's not it is in form of injections. It's not naturally passing from mother to fetus, but it is intentionally introduced artificially through means of injection. Then that kind is an example of artificial passive immunity. Okay, so example of them will be snake anti-venom. So venom of the snake will have proteins that can act as an antigen. So they have to be neutralized by anti-serum. Okay. Anti-venom is nothing but an anti-serum. Okay. Anti-serum. That is serum containing antibodies. Serum containing antibodies is called as anti-serum. I told the last time itself that antibodies will be present in blood, in lymph, but how it can be isolated? It can be isolated in form of serum. Okay. So therefore, snake anti-venom, it is going to be present in the serum of an animal where it is raised. So this anti-serum preparation will have antibody. That itself is prepared in the form of injections and given to a person. Okay. So whether the snake venom is hemotoxic or neurotoxic, they are basically protein antigens. So they have to be neutralized by antibodies. This is one example. Second is anti-tetanus, which is popularly called TT injections, anti-tetanus. This the pathogen cause is serious and dangerous, so it does not give enough time for immune system to produce. So quickly, anti-tetanus injection has to be given. Then third, even for rabies, anti-rabies. So these are some of the examples of which immunity? Passive immunity. So this completes difference between active immunity and passive immunity, broad difference which is very important. Then further you can systematically classify them into natural, artificial, natural and artificial. Now next is the yesterday's topic as a continuation, that is types of acquired immunity. So types of acquired immunity. Already I told there are two types, innate and acquired immunity. Types of acquired immunity. There are two types of acquired immunity. One will be humoral immunity. These are main branches of immunity in our body. So one is humoral mediated immunity, and the second one is cell mediated immunity. In humoral mediated immunity, the immunity is brought about by production of antibodies which are present in body's humors. So here what you are noticing is this one. Which word is supposed to be here? Humor. And last time I told what is a humor? While starting itself, in the introduction of human health and disease, humor is nothing but body fluids. Now, which are the body fluids? The body fluids will be blood and lungs. Main body fluids of our body will be blood and lung. Now, the antibody produced, our own antibody produced, will be present where? Antibodies in body fluid. If the immunity is due to antibodies in body fluid, then that type of immunity will be called as, just a minute, antibodies in body fluids. So if a person is resistant to disease due to presence of antibodies, okay, due to antibodies present in body fluids, presence of antibody in blood organ, then that type of immunity is called as humoral mediated immunity, which is mediated by antibody present in humors. So it's also called antibody mediated immunity. Antibody mediated immunity. On the other hand, word itself, here also word says everything. Here what is mediating the defense? Here what is mediating immunity? Cells are mediating. Antibodies are molecules. But here cells only will directly release substance and attack the target cells. Understand? So cells will mediate the immunity here. So it's called cell mediated immunity. Okay. Which are the cells here? Remember, the main cells will be T cells. In this, there can be type like T suppressor cells, T helper cells, T cytotoxic cells, like this. Got? So these are what you call some of the T cells. Got? So these all T cells. T stands for they get matured in thymus, which you already studied in hormone chapter. Okay, that means the last chapter in the 11th class. So these T cells will mediate this immunity. Due to T cells, the person will not get the disease, not due to antibodies. Okay, so immunity is due to cells and immunity is due to antibodies. Is the main difference here between humoral mediated immunity and cell mediated. TS cells, see here, once the pathogen will get destroyed, it doesn't mean the cells should continuously produce antibodies or cytokines. Now they should get suppressed. The immune system should get suppressed once its work is over. So that role of suppressing an immune system is due to TS cells. Helper and cytotoxic. They kill the target cells. These help both B cells as well as T cells. is mediated by antibodies. Immunity mediated by antibodies. And here, immunity mediated by cells. So this is the difference between humoral mediated immunity and cell mediated immunity. The first difference, then coming to second difference between humoral mediated immunity and cell mediated. Which cells will be there? You should know it is the B cells. Original B cells are responsible for humoral mediated immunity. You should know ultimately B cells produces plasma cells, and it is the plasma cells that will produce antibodies. Okay. So here, this is mediated by T cells. This is going to be mediated by T cells because they mature in thymus. Next, third difference will be humoral mediated immunity is very good for pathogens like bacteria and virus. Bacteria as well as viral pathogen. This is effective against bacteria. Usually, it is good for neutralizing bacteria as well as virus. Which one? The humoral mediated immunity. Humoral mediated immunity. Whereas almost all pathogens can be destroyed by cell-mediated immunity. All pathogens means apart from bacteria, virus, fungus, protozoa, like that. So helminths like that. So all pathogens can be effectively dealt by cell-mediated immunity. All pathogens can be destroyed by cell-mediated immunity. This one is limited in its. This is again the next difference between humoral as well as cell-mediated immunity. The next one. It is not good for having anti-cancer effect. So it is not good at curing the. It cannot usually act on the cancers. So not effective. Or it does not provide immunity against cancer. Does not provide immunity against cancer. What is cancer? Cancer is formation of malignant tumor where the cell loses its control over the cell division. When the cell loses its control over the cell division, it will start to divide continuously. As a result, a mass of cell is formed in the form of tumors. In that, cancer is caused by a harmful tumor called malignant tumor. But humoral immunity is not good in providing immunity for cancer. So this provides to a certain extent, even though cancer is not curable, but immune system is not keeping quiet. It has to respond to cancer formation, to cancer tumor formation. So here what happens? Here it will provide to a certain extent. It will provide to a certain extent only immunity or response. Okay. Provide immunity to certain extent to cancer. So to a certain extent it can still fight against the tumors, but here These are not, this branch of immunity is not good in what you call uh, giving immunity to us. So next will be, but you should know the phenomena of organ transplantation. Now here, two individuals are there. Two individuals are there. So consider he is a donor for an organ and he is a recipient for an organ. Got? So what happens here is recipient's organ is become non-functional. Consider that his kidneys, both the kidneys have failed. Okay. Now you want a donor to donate one of his kidney to him. Understand? So therefore, what happens? It is the transplantation has to be done. So therefore, organ is transplanted from donor to recipient. Okay. So that the donor's organ will start functioning in recipient. Okay. So first thing you should understand, donors, a donor and recipient will be genetically exactly identical. It is not possible. Understand? So what happens? Suppose the organ from the donor is transplanted into recipient. Okay, this is called organ transplant. It's called as organ transplant. Now, recipient's immune system starts considering this kidney that has come from a donor as foreign, and will start responding to it, start attacking to this organ that is, the organ that is. So therefore, in recipient, what happens is immune system starts responding to the starts reaction or transplantation after the transplantation. So immune system starts responding to the graft here. What is a graft here? So this organ transplanted will be rejected here or rejected by the immune system of recipient. So graft rejection it is called. When the recipient immune system starts reacting against this organ, we say that there is graft rejection. Okay, because they are not genetically identical. But if they were genetically identical like monozygotic twins, then immune system won't respond to the grafted organ. Can you understand here? So therefore, immune system of recipient will consider this organ transplanted as foreign and start reacting against it and start producing immune cells to attack these tissues of this particular organ, and finally what it will do, reject the graft, reject this organ. Okay, means what is responding? Immune system. So what is given to such recipient? So that means this organ should at least be in this person for a few years. For that reason, the recipient is given with immunosuppressive drug. Is given with which drug? Immunosuppressive drug, like cyclosporin is an example of immunosuppressive drug. What? So that is given to a person. So immunosuppressive drugs mean these drugs, medicines will not allow the immune system to respond. So that what will remain inside the body of recipient, the organ, will remain without being attacked by immune system, without being responded by this immune system. Understand? But there is a risk of use of immunosuppressive drug is he will be more vulnerable to other pathogens. It is a general suppressive, it's not a particular specific suppressive effect. This immunosuppressive effect will have suppressed the entire immune system, so that immune system in a suppressed state, what happens, it will stop rejecting the graft, but at the same time it cannot respond to other pathogens that are entering the body, so the person will be more vulnerable for infections and other things. So that means immunity becomes weak here. Okay. So that is how the organ that is transplanted is extended for few years further due to use of immunosuppressive drug. So that is called as graft rejection, remember. Now, humoral mediated immunity is not involved in graft rejection. Whereas cell-mediated immunity is involved in graft rejection. So if our body rejects the transplanted organ, it is not the humoral immunity, it is the cell-mediated immunity. Rejection of the graft. Now if I ask, which cells are involved in graft rejection? Whether it is B cells or whether it is T cells. So you should know a little bit of B cells and T cells also. So B cells will be the cell-mediated immunity, that is graft rejection is due to B cells or T cells. You know that graft rejection is due to cell-mediated immunity and cell-mediated immunity is due to T cells. So it is the T cells that are involved in rejection of graft or rejection of an organ. Okay. So and any of the organ cannot be easily managed from one person to another unless there is a unless both individuals are genetically identical. And one more is eye transplant, which involves transplantation, usually the portion called cornea, but cornea do not have blood supply, so the cornea of one person can easily be transplanted into cornea of the cornea replaced. I mean, the cornea of one person can be transplanted onto the another person without a rejection because the cornea do not have blood supply. So usually the WBCs, which are part of immune system, they don't attack the corneal tissue, and therefore cornea transplant is very common. And since these two cells are commonly mentioned, yeah, I'm going to explain about the B cells as well as the T cells. See, B actually stands for burst of fabricaceous, and we can tell here the maturation will be in the bone marrow. The B cells, ultimately the original cells, the stem cells, most of the blood cells will arise from the stem cells. Okay. So B cells actually will arise in the bone marrow and mature in bone marrow. So origin is bone marrow. And here maturation of B cell occurs. Now what is bursa fabricius? That is present in birds. Okay. So birds, the bone marrow is very, if at all bone marrow is present in any birds, it is going to be very negligible and does not have a role in hemopoiesis, that is production of blood cells. So therefore, the bird's protective tissue will be the bursa fabricius. That is, cells of defense are produced in bursa fabricius. So that's why the B. But here the bursa equivalent means here in the comparative to bursa fabricius, that means bursa fabricius, similar tissue will be in our body as bone marrow, where the maturation of B cell occurs. Now T cells, origin occurs in bone marrow. They originate in bone marrow, but site of maturation is different. The site of maturation of T cell is thymus. Site of, note it, site of maturation is thymus. So then what? Another difference is maturation is one difference. Then B cells are responsible for humoral mediated immunity. T cells are responsible for cell mediated. The next difference comes, B cells are not involved in graft rejection, whereas T cells are involved in graft rejection. Now other differences like B cells have capacity to produce antibodies after getting differentiated into which cells? Plasma cells. Whereas T cells will not produce any antibody. So these are the main differences between the two main cells of adaptive immunity or acquired immunity, not innate. These two are cells of acquired or adaptive immunity and not innate. You have to remember. Okay, so these are some of the differences between the two cells of immunity. Okay, so this completes active immunity, passive immunity, humoral cell-mediated immunity as well as cell-mediated immunity, and some of the difference between T cells and B cells. So also you have studied in last chapter that thymus is going, what you should know about this is it is also an organ of immune system. Okay. Further you will study which are the different organs of immune system. So this completes today's topic. Further we will continue with vaccination and vaccines.